Hello students, today we are going to read a very important topic in the molecular basis of inheritance, that is called the genetic code. See, so far whatever we have studied, that is regarding the replication or transcription, it was always the nucleic acid which was converted or rather changed into another type of nucleic acid on the basis of complementarity. But as far as the translation process is concerned, here the nucleic acid has to be coded in the language of amino acids, that is in the process of protein. Although if you talk about, there are enough evidence to prove that yes, if the nucleic acids are resulting into proteins, the reason is because if there is a slightest mutation in the genetic sequence of an organism, that leads to production of a new type of protein or a polypeptide chain. So but it could not be assumed, or it was rather very difficult, and there were no real evidences to properly say that how this that is the nitrogenous base sequence which is present in the mRNA could be translated into that of the protein. So the deciphering of that is your genetic code was a challenging task for the scientist. Now there were various scientists from various disciplines who came forward and he worked together. There were people who came from the biochemistry disciplinary, who came from there were physicists, there were people who were from different backgrounds, and they all came and tried to actually find out that what could be the possible combinations, or what could be the possible way of finding out the genetic code which would be responsible for the coding of the amino acids. And it was the scientist, it was George Gamow, who suggested that it could be so like that whatever the nucleic acids are there, if we just arrange it by means of permutation combination, then since there are 20 amino acids, so if we arrange them in the sequence of since there are 4 nucleotides, if we arrange them in the sense that the 4 to the power 3, that means the 4 nucleotides are, if they are in the arrangement of 4 into 4 into 4, so it is becoming total of 64 nucleotide sequences, or you can say the codes which will be coding for the 20 amino acids. Although these were becoming more because for 20 amino acids 64 base sequences, and then he said these base sequences must be in the form of triplet. It should be in the form of triplet, and then only the process of encoding of the nucleotide into amino acid was possible. He was supported by some scientists, one of the greatest scientists of all time, Herwin Khurana, and Nirenberg. They worked synthetically, they synthesized the protein synthetically by means of yeast, the organism yeast, and they were capable of synthesizing the RNA in laboratory condition using certain enzymes which were also synthetic in origin, and they came to a conclusion that yes, that Gamow was quite correct. That means if the combinations of the 4 base pairs of the DNA could be in the triplet form, then the genetic codon can be derived. And they came to a conclusion by means of these kinds of experiments which they took into the laboratory using some unicellular organisms like yeast, and synthesizing some synthetic enzymes to polymerize those nucleotides and to produce an mRNA, and to produce subsequent amino acids with respect to the sequence of those nitrogenous base pairs in the mRNA. They gave this genetic codon. Okay, so the genetic codon, they said the properties of the genetic code, the characteristics, or you can say the salient features of the genetic code into the following that is the features or the following types. They were of the following characteristics. They said that the codon must be in triplet. It cannot be, in that is the base pairs cannot be in 4 in a frame to be read by the transfer RNA, which will be bringing the amino acid with respect to the genetic code which is present in the mRNA. So they said that the codon must be a triplet codon, and they said that if the permutation and combination which was proposed by that of George Gamow was true, in that case there are 64 possible combinations of the genetic codon. Now out of this 64 possible possibilities of this genetic codon, they said that 61 are capable of coding the amino acids. So what about the other 3s? The other 3 codons do not code for any kind of amino acid, so they are known as the stop codons. Okay, that means whenever the translation is to be stopped, there must be some signal present in the mRNA which will be now letting the stopping of the polypeptide formation, or else what will happen? It will keep on happening. So there must be some termination points. So those termination points is also determined by certain codons, and these were the 3 codons which we call it as the stop codons. Actually they are sometimes known as the nonsense codons. Okay, so because they do not make any sense and there is no transfer RNA with respect to these 3 codons to bring any amino acid, so that's why also they are known as your stop codons or nonsense codons. And these codons are what? These codons are UAA, okay, you can call it UA, okay, UGA, UGA, and UAG, UAG. Okay, so these are the 3 stop codons. They have separate names also. They are known as your ochre, opal, and amber. So these 3 codons do not code for any kind of amino acids, but never a discovery is stopped. So later on, when the people again wanted to verify these points proposed regarding the genetic code, they found that in case of some lower unicellular organisms, the stop codons, some of the stop codons, not all of them, are also capable of synthesizing certain amino acids, but it is not for everyone. Universally, if you consider universally, UA, UGA, and UAG, these 3 codons are the stop codons. They do not code for any kind of amino acids, and the rest 61 codons, they are responsible for coding the amino acids. The next point which was included under the salient features, it includes the codons are unambiguous and specific. That means for a specific amino acid there will be a specific genetic code. That means a specific genetic code is going to only synthesize a particular amino acid. The same genetic code cannot be utilized or cannot be used or cannot be specifying another amino acid. It cannot be used to specify another amino acid. So it is just 1 to 1. So that means one amino acid, it will be that is your decoded, or that will be brought about with respect to a particular genetic code. So one genetic code, one amino acid. Okay, the next point is codons show degeneracy. Now this is just in continuation with this. That means here we have said that codons are specific or unambiguous. That means for a particular codon, one amino acid. But it does not mean for one amino acid there is one codon, because there are 64 combinations and there are 20 amino acids. So for one amino acid there can be more than one codon, but for one codon there is one amino acid. So for one codon, one amino acid, that is point number 2, unambiguous and specific. Sometimes this question comes, what do you mean by unambiguous? You have to explain it. That means for a single codon, a single amino acid. Okay. And number 3, codons show degeneracy. Degeneracy of codons means what? That is, there can be more than one codon for a specific amino acids. Okay. Next, number 4, they are read in mRNA in a continuous fashion. Okay, in a contiguous fashion. There is no punctuation. That means it is not that there will be any stopping in between or any pause in between the respective codons. That means suppose there is U. Okay, then AUG, okay, UGG. So all will be in continuous fashion till the termination codon is reached. It won't be that it will be stopping for some specific segment of this before the termination codon is there, or it will be utilizing the previous nucleotide base pair and the next of the frame. What is this frame? Is that means a triplet. Codon is a triplet, so there will be 3 nitrogenous bases, always 3 3 3 3 3 like this. It won't be like 2 of the first, next 3 of the second, next 3 of the third, like this. No, it is always in a contiguous fashion and always in triplet form. That means it will be reading the frame will be shifting with respect to 3 nucleotides in an mRNA. Okay, so that is the meaning of the 4th point. The 5th point is that the codons are universal. This is very important. That means whatever be the codon, that is the nucleotide sequence for that of bacteria synthesizing or is responsible for an amino acid, will be same for human beings also. Okay, that means suppose for example say for example the genetic code say UUU. Okay, now UUU is a genetic code which is responsible for the bringing up of the amino acids called phenylalanine. Now this UUU will be same for bacteria also, it will be same for us also. So that is the meaning of this, that the codons are universal. For all the living organisms on this earth, the codons are universal. Exceptions are there. What is the exception? In case of human beings, you know, there is an organelle which is called mitochondria. In that mitochondria also there is a DNA. So if that DNA of that mitochondria or the mitochondrial DNA, in that case the codon may not be universal. That means the codon which is responsible for the bringing up or synthesizing of a particular amino acid may not be responding with respect to the mitochondrial DNA of the human being. Okay, so those are few exceptions, but mostly right from bacteria to the highest means evolved organism, that is mammal, the human beings, the codons are universal. Whatever they will be showing the effect or are responsible for the amino acid in case of bacteria, it will be same for us also. The last point is that AUG has dual function. Now this codon AUG is a dual function codon. Dual means what? It can perform 2 functions. Now what is the 2 function? It is the initiation codon. Initiation codon means the start codon. Any stretch of the structural gene which is to be synthesized by the cell during the time of its need always begins with this AUG sequence in the mRNA. So it is the start codon or the initiator codon, and sometimes it will be it will be actually synthesizing for methionine. It is responsible for bringing up of the amino acid. It will be coding for the amino acid, rather methionine. But there is another codon also, that is your GUG. Okay, now that GUG is also sometimes acting as a initiator codon. Okay, so that is always not there, but this is always being found that it is responsible for the starting up of the translation process, as well as it is also responsible for the synthesizing or for the bringing up of the amino acids or for coding the amino acid, which is called the methionine. So these are the various 6 salient features which were proposed by these eminent scientists of all across the world, and thus the genetic code was being deciphered, which was a long time a very challenging task for these scientists, and that is how the process of translation could be understood in view of the various kinds of codons which are responsible for the synthesis of 20 amino acids. So it was ultimately 64 combinations of the nucleotides which was responsible for the decoding or for the that is your synthesizing of the polypeptide chain with a combination of the 20 amino acids present in the cell cytoplasm. So that's it about the genetic code. The next topic which is discussed in your chapter is about mutation. Now just now we were discussing about this point, that is number 4 point, that they are read in the mRNA in a contiguous fashion. That means triplet codons are responsible for decoding of the amino acids. One single amino acid will be coded by a triplet codon. That was the meaning of this point number 4 in the salient properties of the genetic code. Now regarding mutation, you have a previous idea that whenever the cell is subjected to some kind of mutation, some kind of agents which are responsible for mutation, then that results into change in the base sequence or the genetic sequence of the organism. And as the genetic sequence of the organism is changed, the base pairs are changed. In response to that, the usual protein which was produced earlier prior to mutation, it also changes. So with that also scientists were quite evident, or they were quite assured that yes, the nucleotides sequences are only translated in the form of amino acids. So if a mutation happens with respect to a single base pair, which we have seen in case of your sickle cell anemia. In case of sickle cell anemia, perhaps you remember that in the 6th position, instead of the glutamic acid, which was the amino acid, if it is replaced by valine, okay, so in that case what will happen? This will result into a faulty beta globin chain of the hemoglobin, and as a result the shape of the RBC changes, and that may result into oxygen deficiency carrying capacity, and that may lead to various kinds of phenotypic changes in the organism that we have seen. So what happens in case of that point mutation is a single base pair is changed. If it is, suppose a particular codon is only removed, that is called deletion, or it is being introduced, or it is being inverted, which is called inversion, or it is transferred to some other location, translocation. These all results into various types of mutation in an organism. But always we know that the frame of the nucleotide sequence should be maintained. That means 3 codons at a time. In your NCERT book, you will find that they have explained you with respect to a particular sentence, Ram has a cap. Okay, Ram has cap. Okay, for example, Ram has cap. Now in this sentence, Ram is in 3 alphabets, RAM, has, HAS. This is also 3. So you can take it to be the example of the codons, triplet codons. Ram has cap, CAP, cap. So that is also a triplet codon. Now in this sentence, Ram has cap, if I introduce after Ram has R and then cap, then what will happen? We all know that nucleotide bases will be in triplet form. So Ram has is up to has is okay, but after that, since I have introduced one capital R in it and cap is there, so in this cap, RCA will be together and P will be separated out. Understood? So since because it will be contiguous and it will be in the form of triplet, that is why the last alphabet is alone. Okay, now if in the, suppose in the second mutation it is like that, RE, then what will happen? Ram has is okay, then REC, okay, and AP will be separated. Okay, because it has to be in contiguous form. Then in the next stage, if suppose a D, a new nucleotide base has been introduced, so Ram has red cap. Now you see, these are the 3 triplets. Ram is also 3, has is also 3, okay, red is also 3, cap is also 3. So this way the frame of the nucleotides, that is the triplet codons, changes if there is any addition or deletion of any base pair within the, that is the sequence of the DNA, which has to be of course transcribed and then translated. And as a result of the change in the frame, that is sequence of the nucleotides, which is in triplet form, this results into mutation, and this type of mutation is called frame shift mutation. Because you see, in this case what happened, in the beginning I said Ram has cap. So in that, when I am using R, see the frame includes the 3 nucleotides. So Ram has, these 2 frames are same, okay, R cap. So R cannot be alone because nucleotides has to be contiguous. So RCA will be together, and P will be separate. So frame of cap has been shifted. It has been shifted, it has been changed. So as a result of changing that, a new amino acid will be synthesized, and that will be, affecting the phenotypic nature of the organism. So deletion or inclusion, that means something which is included. So that will result into the changing of the frame of the nucleotide codons which are present in the DNA, resulting into a new formation of a polypeptide and resulting into change of the expression, phenotypic expression of the organism. So that is about the mutation and how the mutation affects the phenotypic character of an organism. See, in the next classes to come, we are going to read about the translation process, and after the translation there is a very important thing which is called the gene expression. Now this gene expression, if you have seen in the previous classes, in that I have said that gene regulation is at various level in case of eukaryotes. The reason is because you see that the formation of the DNA from DNA and the formation of mRNA from DNA, all this takes place in the nucleus. So this is one type of, you can say, regulation. Then what happens, the mRNA which is produced, now that mRNA in case of eukaryotes, when it is just produced, it is called heterogeneous mRNA, because it is containing the introns and the exons. So the introns are the non-functional genes, so it has to be spliced. So when the splicing is done, that is also a kind of, regulation, isn't it? And after the splicing is done, then again it is joined together, the exons are joined together. When the exons are joined together, that is also a kind of regulation. After the exons are joined, then processing is there, that means the capping and the tailing is there. So there is also a regulation. And while the movement of this mRNA, the ready mRNA from the nucleus to the cell cytoplasm taking place, there also some kind of regulation takes place. There are some, there is a possibilities of certain enzymes to be acting upon it. So there also it needs to be, taken care of, so that whatever is the required result, it is not hindered because of the effect of any other enzymes on it. So regulation is at various levels. Then while the translation is happening, that time also the regulation is there. So various levels of the regulation is there, which we will be seeing it in case of the organism, in case of prokaryotes, like in case of organism like E. coli, where we will be seeing that how the operon model is there for the regulation of the gene expression, and in case of eukaryotes, how does it happen. And then we will be taking upon the human genome project, and finally the DNA fingerprinting. So today's class is up to this much, that is the genetic code and the salient features of the genetic code, plus mutation and how does the frame shift mutation takes place in case of the DNA.